Hello everyone. In this video we are going to continue the groups of the invertebrates. So now we will start in the bilateral, and the first one is the Lophotrochozoa. So we have examples of the Lophotrochozoa, the flatworms, the Mulaskas, and the Analytes. So starting with the Platyhelminthes, and Platyhelminthes means flatworms. So platy means flat, and helminthes means worms. So these are flattened dorsoventrally, and they have a gastrovascular cavity. So since they have gastrovascular cavity, that means they are neither proto nor deuterostome, because being proto or deuterostomes refer to the presence of a mouth, and whether the mouth has been developed first or second. So here we don't have a true mouth. That's why it is just a cavity serving the two functions of being like a GIT and as well as a circulatory system. So the platyhelminthes are characterized by being bilateral. So moving on from the previous two groups, if you remember, the sponges and the cnidarians, now we started to the bilateral animals. So bilateral means that they have head now. Also they are tripoplastic, meaning that we have now mesoderm, and in terms of the cavity they are coelomate, so they have no cavity. And actually most of the functions within the platyhelminthes comes from their names being flat. So gas exchange take place through the surface, so the surface of the body of the animal is very thin, allowing most of the functions to happen by diffusion. They have also protonephridia, which is a primitive organ similar to the kidneys that we have, in order to regulate the osmotic balance and to avoid that they suffer from osmotic stress. So here is a figure showing an example of one of the platyhelminthes, and what we can see is the presence of the head. So in the head we have accumulation of ganglia, so accumulation of neurons, and they are accumulated here in the centralized region. Also they have eye spots. So eye spots, they are light sensitive. They don't allow processing of images, but they allow the sensitivity of light, and through which the platyhelminthes, they detect the source of light, and they interact or respond and align themselves towards the source of light. In terms of the digestion, how they deal with that is through the pharynx. So they have this pharynx, and what they do is they secrete digestive juices out of the pharynx. So let's imagine that here we have food source. So this secreted juices will go digest this food source into smaller molecules that will be absorbed through the pharynx and will be transported through the gastrovascular cavity. So what is shown here in brown is the gastrovascular cavity, and as you can see it covers a very large surface area, especially with this convolution, and the digestive food molecule will be further digested in this gastrovascular cavity and will be distributed among the different parts of the body surface. So moving on, the second phylum will be the Mulaska. So they are characterized by having a soft body, but most of them, they are protected by a hard shell. So for example, we have the land snail, we have the sea slug, the octopus, the squid. So again, they are bilateral, tripoplastic, and here we have a true coelom, and the mouth has developed first, so we refer to them as protostome. The body plan of Mulaska contains three main parts. So they have the muscular foot, which is shown here in like light brown color, and you can imagine like the snail, so they help in the movement of the organism. Then we have the visceral mass, so all the internal organs, and as you can see we have a digestive tract that starts with mouth, and the mouth has evolved first. Then we have it ends with the anus, and it has a circulatory system and nerve system. So all these internal organs are collectively known as the visceral mass. Then we have what is known as the mantle, so which is shown here in blue. So this mantle secretes the shell. Remember, they are covered by, most of them covered by a hard shell. So this blue structure, the mantle, will secrete the material that produces the hard shell of the organism. How they get their feed? This is done through a rasp-like radula. So as you can see here, if we zoom in at the mouth area, near the mouth area, they have this rasp-like radula. So they are very rough structures that scrape the surface of, for example, the bottom of rocks, where they remove algae from the bottom of the rocks or from any other substrate to get their food, these food, and then they absorb it through the mouth. When we move on to the higher mollusks, for example the octopus, these are characterized by having a very good eyesight, so precise vision, and also they have very good problem-solving abilities. So we usually tend to focus on vertebrate when we talk about problem-solving abilities, but some of the higher mollusks, they are very intelligent. And here a picture where somebody has given this octopus a food source in a capped bottle, and as we can see, the octopus has opened the cap in order to reach the food. The last group that we'll focus on is the Analytes. So Analytes, they have bodies composed of series of fused rings, so they are known as segmented worms. So again, they have bilateral symmetry. They also have the mesoderm, so they are tripoplastic. They have true cavity, they are coelomate, and the mouth has developed first, so they are characterized by protostome development. In this slide, we are showing the importance of having a true coelom. So Analytes are coelomates, they have true coelom, and this coelom is shown here. So it is the cavity. So this cavity is the coelom, and the importance of that, as you can see, the Analytes, they have intestine, they have circulatory system represented here by this dorsal vessel. They have also the protonephridia, they have primitive nervous tissues, and all of these organs, they sometimes expand. For example, imagine an intestine. It expands, it needs to move the food, and all of that needs space. And in addition of that, the coelom also allows the tissues to differentiate more, so allowing each organ to work or expand without the need of the movement of the rest of the structures. And in addition to that, it allows better differentiation of the tissues as well. So if this Annelids was a coelomate, that was not possible, because each time the intestine would move, that would cause the movement of all the other tissues, and there is no space for expanding or further differentiation of tissues. Leaving the Lophotrochozoa and now moving to the Ecdysozoa. So Ecdysozoa are covered by a hard cuticle that is molted through a process called ecdysis. So this hard cuticle is not flexible. It is not allowing the expansion of the organism. So in order for the organism to grow in size, that needs shedding or casting off the old cuticle, and then new bigger cuticle is formed to allow the growth of the organism. The Ecdysozoa are also bilateral animals, and the two largest phyla within the Ecdysozoa are the nematodes and the arthropods. So if we move on to this slide, you will see that there are different phyla, but the biggest one in terms of the number of species would be the nematodes and the arthropods. As a side note, this group of organisms, the tardigrada, is very important. So sometimes they refer to as water bears because of their shape. They show very characteristic survival abilities. So it has been shown that they resist so they have been kept in nearly boiling water and they are still alive. Another experiments have shown that they resist living at very high pressure, so which is nearly 6 times the pressure at the deepest ocean. And in another part of the experiments, it has shown that they have been able to withstand living for 10 years without water and food resources. So they resist desiccation. So they are very interesting because of their survival abilities. So moving to the two phyla with the highest number of species, so we have the nematodes. Nematodes are the roundworms. They are important decomposers in the soil and the sediment of the soil. Many of them, they are parasitic. Again, they are bilateral, and they have the mesoderm, so they are triple plastic. But in terms of the cavity, they are pseudocoelomate. So pseudocoelomate, to revise this word, we mentioned that that meaning that the cavity, the coelom, will be covered with the mesoderm from the outside and the endoderm from the inside, and they are protostome in terms of development. So moving to the next group, we have the arthropods. This includes the insects, crustaceans, spiders, and they are characterized by having segmented bodies with jointed appendages. They have hard exoskeleton made up of chitin that provides protection and attachment points for the muscles. And as we said, what is happening is that the exoskeleton must be shed for the growth. So what is happening is that the old exoskeleton with the chitin, so the hard molecules giving the old exoskeleton its hardness, will be absorbed by the organism. So most of the chitin will be absorbed. The remaining ones will be shed out. Then new exoskeleton, new bigger one, will be produced, and then it gets hardened by time, and by doing that, the organism is allowing to grow in size. They are bilateral animals. They have the mesoderm, so triple plastic, and they have true cavity, and they are protostome in terms of development. Thank you so much.